Hello, everyone, and thank you so much for joining us today. My name is Jin Chiu, Director of Marketing at PackGene's. I'm very happy to welcome you to the exciting webinar. Before we dive in, let me briefly introduce PackGene. So we are a global CRO and CDMO company specializing in customer AAV production as well as antivirus, mRNA, and plasmid DNA manufacturing. So our mission is to accelerate gene and cell development by providing high quality and scalable products and solutions. So we support our partner from early discovery all the way through clinical and commercial stages, helping them bring therapy to patients faster. So today's webinar focus on an important topic, the dual-plasmid system for AAV packaging and production. As many of you know, traditional triple-plasmid system has been the key standards for more than 2 decades. It is effective, but it also comes with a lot of challenge. In the recent years, there have been growing interest in developing more efficient and less costly alternatives. So we are very excited to share PackGene's latest data and innovation with our dual-plasmid platform. So with that, it's my pleasure to introduce today's speaker, Dr. Amos Garnick. Amos brings a decade of expertise in molecular biology and gene therapy. He earned his PhD from Weizmann Institute of Science and completed postdoc training at Harvard Medical School. Previously, he led preclinical gene therapy research at Biogen and PackGen. So today, he will guide us through the design, the data, and the performance of our dual-plasmid system. So if you have any question during the talk, please feel free to type them in the middle speech bubble icon you will see in the upper left corner of your auditory page. We will gather all your questions and address them during the Q&A section at the end. So with that, I will hand it over to Amos.
Thank you, Jin. And thanks, everybody. Good morning, afternoon, evening, wherever you are. It's an amazing couple of weeks to be here. I'm sure many of you have heard the news last week that the Huntington's disease community received. It was the best news they've had in decades. For the first time ever, gene therapy was actually able to slow significantly progression of Huntington's disease in clinical trials. And this really showcases the incredible potential of AAV gene therapy. Well, so why is it so rare? There are a lot of genetic diseases. Why is it so hard to run a gene therapy program? And I think we all know the answer. Unfortunately, it's cost. It typically takes between $500 million to $1 billion to develop a breakthrough like this. And even just one dose can cost as much as $500,000. And these sky-high expenses are the main barrier, or one of the main barriers that prevent these promising therapies from reaching patients. So PackGene was founded about just over a decade ago on a single simple tenet, and that is our company motto, which is make gene therapy affordable. And our mission is, as Jin said, to break down these cost barriers and help bring life-changing treatments like the Huntington's therapy and others to families and patients around the world. So yes, we are a global CRO CDMO. We're focused on gene therapy. We have end-to-end services from plasmid construction, AAV manufacturing, a track record of over 10,000 projects, well over 1,000 customers worldwide, from small, scrappy academic labs in the top institutions to big multinational corporations. So since our founding in 2014, we've been working to make gene therapy more affordable by offering scalable, high-quality AAV and plasmid platforms and incorporating innovations, technological innovations, basically at every level of our production pipeline to increase yields, lower impurities, reduce costs per batch. And all of these incremental improvements in our pipeline, they add up to a big difference when you're trying to take a gene therapy program up to scale to the clinic. So excitingly, recently, 2024, we launched our brand new 35,000 square foot GMP facility here in Houston, Texas. It's fully operational now, global regulatory standards. It's producing currently GMP materials for current trials, for clinical trials. So we really are offering services around the globe. So our services, as I mentioned, and we'll talk today, our mainstay is the AAV. We produce GMP plasmid. We make RNA and LNP formulations and also lentivirus packaging. But today we're going to be focusing on our AAV services, which is the heart of our innovations. So I'll discuss today, I'll break it down to the three core platforms that allow us to pursue this goal. These are our Pi Alpha AAV high yield platform. That's most of what we'll be talking about today. Our Pi Omega plasmid DNA high yield production platform, and also touch upon our exciting Pi Icosa AAV capsid engineering platform at the end. So for the most part, we'll be discussing our real flagship innovation, which is the Pi Alpha AAV high yield platform, and the dual-plasmid platform that is our newest innovation in this system. So for all of you that are very, very familiar with all things AAV, I hope you will indulge me a brief moment. I'd like to quickly get everybody up to speed on AAV 101 and just start by describing the natural life cycle of an adeno-associated virus, right? When there's no helper virus present, the AAV enters the cell. It has rep genes and cap genes in its viral genome, flanked between its inverted terminal repeats. But even though it gets into the nucleus, uncoats, and becomes a episome in the nucleus, it cannot express the rep and cap genes. It can't overcome the natural defenses of the cell. And in this silent state, it limits the virus's ability to spread or to infect. Now, of course, when a helper virus like this adenovirus enters the cell, everything changes. The co-infecting helper virus provides vital helper functions by expressing a host of helper genes. And these genes turn on the dormant AAV genome. They allow rep and cap proteins to be produced, the genome to be replicated through the ITRs, an assembly of new AAV particles in the nucleus, and eventually the cell undergoes lysis. And now we have these new infectious AAV particles. So let's just compare that with how we use recombinant AAV as a tool for gene delivery and therapy, in gene therapy. So in the lab, we engineer the AAV to carry a therapeutic gene of interest, and it's bound by specific ITRs. And we replace the rep and cap genes and put in our own gene of interest, in this case, a simple promoter with a transgene. So this capsid can now infect the cell, and our gene of interest can integrate into the nucleus, become an episomal DNA, and express our gene of interest indefinitely. So how do we make a recombinant AAV? So what we need for this is the same things that the natural AAV needs. We need the helper genes to be expressed in this cell. We need the rep and cap genes to be available for expression as well. And we need a genome, in this case, our transgene, flanked by the ITR sequences. And when we have all these three things in the cell, then the cell can produce effective recombinant AAV. And so in order to do this, we simply need to transfect the cell, in most cases a HEC293T cell, with a source of these three elements. And that is the basic architecture under that belies the generation of recombinant AAVs, is the three-plasmid system. We have a plasmid that encodes for the helper genes, a plasmid that encodes for the rep and cap genes from the AAV genome, and a plasmid that encodes our own gene of interest flanked by the ITRs. And when we transfect transiently a packaging cell, usually a HEC293 cell, with these three plasmids, then it produces our recombinant AAVs. So that seems fairly simple. So why is this process actually so expensive? So for one thing, yields are very low. Comparing to other modalities, AAV production results in pretty low viral yields per batch. So if you compare that to a program that's doing small molecule or biologics, there's a lot has to go in to just get a small amount of functional AAV. Scalability is in no way simple because we have the triple transfection. We need the right stoichiometrical ratios. There's variability between batches and need to optimize each time in order to scale. So it's not simple to scale this system. Transfection reagents. High efficiency reagents, especially most of them that we use are proprietary, right, in the lab, but those actually, when you go up at scale, are very, very expensive. You don't usually just buy whatever's available on the market. You have to find something that is highly optimized and makes sense from a manufacturing cost perspective. And then finally, and I think maybe most importantly, we need not only the AAV to be GMP quality, the plasmid that goes into manufacturing this AAV has to be GMP material. And making this plasmid is very expensive, especially if you're using 3 different plasmids for this transfection. This is going to count to up to 40% of the upstream production cost in the process. So when PackGene was launched, we brought forward a significant set of technological solutions that allowed us to make initially a better, more effective rep-cap plasmid. And that's the heart of our high alpha, high yield production platform. So the initial breakthrough technology was a proprietary non-coding regulatory element that we have in our novel rep-cap plasmid, which increases yield significantly across different serotypes, different genes of interest in different cell systems. And beyond the plasmid engineering, we've systematically optimized transfection, engineering, harvest parameters, and this has resulted in 10-fold yield improvement. And as we can see here, 76 to 92% full capsid enrichment post AEX chromatography. And this is robust across serotypes. So in this case, we're integrating the molecular design with our process development to really maximize the yield, but also the product quality, which is increased in this case. And most significantly, maybe, is that it's fully scalable. So we've managed to generate a seamless process transfer from 3-liter all the way to 200-liter batches. And we've manufactured single batch yields of 1E17 viral genomes. Residual host cell DNA is kept at industry-leading low levels, and this ensures consistent quality from preclinical all the way to clinical manufacturing. So Pi Alpha system has overcome many manufacturing obstacles, but more recently, in order to move the needle further, we've actually gone back to take a second look at the basic paradigm that underlies the production of AAVs, and that's the triple-plasmid system. So this system has been ubiquitous over 2 decades. It's the workhorse of AAV production in mammalian cells. It's reliable, it's adaptable to different serotypes, and it has proven scalable qualities into hundreds of liters of culture. But it does, as we sort of mentioned earlier, it has several significant drawbacks. The high cost, primarily. Manufacturing 3 different GMP-grade plasmids at large scale is very expensive. Complexity, increased regulatory complexities in terms of regulation of the genes, and potential for batch-to-batch variability due to the need for precise optimization. So it's kind of a juggling act to find the optimal protocol for all 3 of them. And importantly, there are impurities and replication-competent AAVs, which are detrimental and a safety risk. Because we have 3 plasmid, the increased frequency of recombination can actually lead to the formation of replication-competent AAVs. If the rep-cap genes manage to somehow recombine in between the ITRs, then that's definitely a detrimental effect that we don't want. And finally, limited efficiency. We actually see that the yields plateau below what we would calculate to be optimal levels, and this constrains the supply, especially for high-dose therapies, like things that should go into the CNS. So there have been reported already for a while now the idea that the RepCap and the helper genes could potentially be incorporated into the same plasmid to create a dual plasmid system for AAV manufacturing. And this has been published on, and it's been shown to be possible, at least on the bench. But, like we said earlier, a lot of things work well on the bench, but performance can drop or it's not sustainable when you try to scale that up to a manufacturing scale. So at PackGene, we've tested and we've utilized learnings from published work and internal screens and experiments to develop a dual plasmid system that actually works in the real world setting that we can scale up and use for manufacturing. So replication-competent AAV formation is reduced by relocation of the P5 promoter, and that's been published on. So we move that away from the rep genes and integrate it with a new non-coding regulatory element that came out from our own screens. And by that, we've able to significantly reduce the risk of replication-competent AAV to below our actual detection threshold. Now, in order to optimize, as I showed you earlier with the pi alpha system, we systematically use statistically significant DOE optimization protocols in order to find the right protocols, the right ratios of parameters like which transfection reagent would work best, how much of this transfection reagent, at what cell density, and with what DNA amount. So by plotting out the relationship of all of these parameters, we're able to actually come up with optimal yield and to come up with optimal functionality of our two plasmid system. So in this slide, we can see how our dual plasmid system matches up to a more standard three plasmid system. So here we encode for AAV9. For our gene of interest is a GFP plus a luciferase in all these cases. But in groups 1 and 2, we're using our dual plasmid system that I just showed you, and 3 and 4 is our triple plasmid system as we've known it. And our high efficiency system outperforms the traditional 3 plasmid process. We actually see higher yields. We see half the amount of residual host cell DNA, and we maintain the vector purity and the genome integrity. If you can look at the capillary, the EC results on the right there, we see that the genome integrity is the same whether or not we use a dual or a triple system. So we've simplified the manufacturing, but we've actually enhanced our yield and regulatory compliance. Now, importantly, this goes beyond AAV9 or GFP and luciferase. We try out different serotypes. Some of them are harder to manufacture, like AAV2 or AAV8, different genes of interest. And consistently, we see that our dual plasmid system is up to the challenge, even though we've incorporated the RepCap and the helper genes into one simpler plasmid. Remarkably, when we actually tried to use the system with novel, hard to manufacture capsids, serotypes, we found that taking those cap genes and integrating them into our established optimized dual system actually increases the yield. So just because you've developed a novel capsid that is very hard to manufacture doesn't mean that it has to be that way. Potentially integrating those novel capsid genes into the dual system can improve the manufacturability significantly. So this bears the question, if a dual plasmid system is better than a triple plasmid system, then why not just integrate everything into a single plasmid? And this has been reported on. It's certainly been tried. And at PackGene, we don't let any new approach or technology go untested. So we set out to empirically test if potentially putting everything into a single plasmid would offer some benefits beyond what we've been seeing with the dual plasmid system. So in a controlled experiment with the same gene of interest, same cell type, same serotype, we tested 2 versions of our dual plasmid system against our triple plasmid system and against 5 different versions of the single plasmid system. And in all cases, the single plasmid system was not able to exceed the yield that we reached with our dual plasmid system. And I think if you look at these numbers, you can almost see why. There's part of the optimization is also getting the ratios between these plasmids. And when you have everything in one plasmid, you have no degrees of freedom to work with to optimize this system. You can put in more or less of that single plasmid. We put half the amount or almost twice the amount. In either case, we can't actually break that ceiling. It plateaus in terms of the actual titer that we can establish. So from our experience, from our empirical data, a dual plasmid system gives us the perfect sweet spot between having lower costs, less plasmids, but maintaining the degrees of freedom that we need to optimize our system. So, like we said before, the real test is the hardest one, which is scalability. A lot of things work in the lab, but does it work in a bioreactor? And we were happy to see that, yes, in our case, we can scale this up to a system that's clinically relevant without compromising the purity. We have residual host cell DNA and plasma DNA. They're below the process-defined tolerability limits. Our full empty capsid ratio is actually improved if you see the peaks on this analytical ultracentrifugation. The peak on the right is our full capsid that has a fully functional genome, whereas the one on the left is the lighter empty capsid. So definitely this system is one that we have a lot of high hopes for in terms of its ability to bring gene therapy to the clinic and to patients. So I've shown you what we've done with our novel RepCap helper TP11 plasmid. We've also been working on the backbone of our gene of interest, our transgene plasmid, in order to get whatever optimization we can out of that. So we call this our pi vector backbone. And this is something, if you go online on our website, there's some very useful tools for either plugging in annotations and genes that are already out there, or you could put your own in order to design the vector with our pi vector backbone. And we see that our pi vector backbone, as we have it today, gives us higher yields, and actually we get much lower residual plasmid DNA in our preps. So going forward, this we think that this is going to be a valuable addition to our technology. And we see that in vivo as well, it works very well, comparable to control vectors, and at some time points we actually see better expression. Okay. So I've talked about the plasmids that we've developed in this dual plasmid production system. I mention again our DOI optimized transfection, which goes for the process. It's optimized for our dual plasmid production system. I haven't mentioned, but a lot of work has gone into finding and developing a novel monoclonal producer cell line that works very well with this process. Seamless scaling up to 200-liter bioreactors, optimized yield and purity methods, as well as a comprehensive quality assurance suite, over 40 analytical assays that we test the purity and the quality of our AAVs, including we've recently brought online a mass spectrometry instrument for a fast and very accurate and much more affordable way to determine the full empty capsid ratio. So this is a service that we provide. All right, so that's our Pi Alpha AAV high yield platform. I'm just going to touch upon our Pi Omega plasmid DNA high yield production platform because it is, of course, relevant. We talked a lot about the cost of manufacturing the plasmid. Our Pi Omega plasmid production platform, by similar methods, engineering our backbones, engineering our ITRs, we've actually been able to improve the yield of the plasmid production by 2 to 4 times, and this is scalable up to 100 liters. In order to purify these plasmids, we've actually developed and optimized a 2-step purification. So instead of using a 3-step process with these 3 columns, we used a 2-step process with a hydrophobic interaction chromatography column in order to purify the plasmids. And with only 2 columns, we achieve lower residual host cell DNA and higher recovery, and that reduced the cost without compromising the quality. And it's important to note that this advantage in recovery is even more pronounced when what we're running through the column is big plasmids. So it's actually essential, and it goes hand in hand with our dual-plasmid transfection system, because if you recall, that plasmid is somewhat bigger. So again, we have different systems that complement each other in order to reduce the costs. Finally, I'm just going to touch upon our Piacosa AAV capsid engineering platform, because it's very cool and it's very exciting. We've used AI to and continue to screen through and engineer novel AAV capsids, enhance tissue targeting, detargeting the liver, and for us, always important to keep in mind AAV capsids that are highly manufacturable. So these advances, they reduce the required therapeutic dose that you would use, they lower toxicity, and they support safer and more effective gene therapies. But this is, of course, a topic for another webinar. So to sum up, at PackGene, we don't just focus on the manufacturing, we really support the entire gene therapy journey from the beginning, from early development, and we have our eye towards manufacturing throughout that whole process. Our GMP facility in Houston is ready to support clinical and commercial production. We have state-of-the-art GMP clean rooms, dedicated process analytical development labs, QC, warehouse. It's fully operational. And in general, both of our facilities utilize industry standard single-use bioprocessing, serum-free hex suspension cells. We are ready to receive and produce the next generation of AAV therapies. So I want to thank you for your attention. We're very excited to collaborate and to help advance your gene therapy programs. Jin will read out some questions. We'll try to answer some questions, but if your questions aren't answered today, or if you think of anything later, please just reach out to us. We're always happy to discuss and to talk about AAV gene therapy. All right, thank you very much. So I have a question here on how a single plasmid system for AAV production actually works. It would work the same in terms of having all the elements there, but the main problem would be that you would be locked in terms of the stoichiometry. You don't have a lot of freedom except in how you design the plasmid. So first of all, you would have to re-optimize every time you change the gene of interest, and because you don't know how that would affect the behavior. And second of all, the ratios might have to be tweaked cloning wise every time. So that would be potentially a limitation. I see another question here. What kind of yield improvements can we expect with TP11 compared to a triple plasmid system? So consistently, we get between 1.5 to 3-fold higher lysa titers. And for tougher serotypes, and this is important, like A5 or 8, we actually see bigger gains. So it seems like this system is really going to be very helpful for those hard to manufacture plasmids. I have another question here. Let me just read this. So since the system modifies the RepCap construct, does this proprietary change affect the tissue tropism or the long-term immunogenicity profile? So yeah, that's obviously important. You want to make sure, since now we have this proprietary RepCap, that we haven't messed with the actual serotype, with the capsid itself. And the answer is, of course, that no, since we don't modify the cap genes or the rep genes themselves, the serotype remains exactly the same. The only thing that changes is the way that we manufacture it compared to how we would how would you ensure that a replication-competent AAV virus is not produced while repcat genes are on the same plasmid? So this is precisely the reason why a dual plasmid might be slightly better than having 3 plasmids swimming around. There is potentially hotspots that you would need to, that we break up, and part of that is why we moved that P5 and embedded it in a regulatory, non-coding regulatory element to mask some of the hotspots between that promoter and the rep genes. But you also have, the more DNA molecules you have, given secondary structures and the ability to just find random non-homologous recombination, having 2 plasmids seems to reduce the number of recombinations that might produce a replication competent AAV. So in that sense, a dual system is actually more safe than the normal triple system. So here's how much cost does the dual plasmid system reduce as opposed. So yeah, obviously that's going to depend on per case, but in general, we find that we have 2 to 3-fold increase in yield. That lowers the cost. And then the system reduces the risk of batch failures because it minimizes the replication competent AAV, like we just discussed, lowers impurities. So given all of that together, we find that we could reduce the cost by even as much as 50%. Of course, that program specific, but that would be sort of the ballpark. Oh, hi, Jin. Are we at time, or do we have time?
Yeah, so I think a couple more questions the audience submitted. So the other question is, for a company with an existing therapeutic candidate already being produced with the triple-plasmid system, so what does the tech transfer process to PackGene dual-plasmid platform looks like?
Right. So I would say that when we apply our pre-optimized process with the identified, we already have the optimal transfection reagents and the conditions. So we have a proven track record of process transfer into our system. Our system is optimized, and so basically our process development team would work very closely with the customer and migrate that into our optimized system. And as we saw, there was a graph there with some novel capsids that often results in higher yield than what you normally would see before using our system.
Yeah, that's correct. Another question the audience also asked, so how does a TP11 like dual-plasmid perform with a larger, GOIs, or, even the, thinking about more recently, people use the dual play-row vectors for AAV. So how do we do with that?
So in general, we see better yields from TP11. Of course, again, it's a case-by-case basis. Like if you stretch the limit and you go closer to or even above 5 kilobases, then you reduce the yield and the genome integrity. But we find that using our pi vector backbone and our optimized system, it actually helps stabilize these challenging constructs. Of course, if we do if we split it in 2, then that should be less of a problem. But definitely, we gain some stability with our current tools.
Okay. I think the last question, I think this is the last question. So how does the TP11 fit into the PackGene current manufacturing capacity or capability, and what the future development can we expect for the dual-plasmid system?
Yeah, so I mean, we're very happy with the data that we've been seeing, and it's already fully integrated into the Pi Alpha platform. So we already have ready for customers, a complete scalable solution, going from research all the way up to GMP manufacturing. We have ongoing work that's focusing on expanding the pi vector compatibility, more plasmid engineering, adapting P11 for newer capsids that are coming out, and continuing to get these performance gains and, higher yield. Like we said, every small difference at the lower scale, higher yield, higher purity, lower manufacturing costs, all of those add up to very significant manufacturing savings in terms of manufacturing costs when you scale up to a clinical program.
Okay, that's great. Thank you, Amos. I think this concludes our Q&A section, and I want to thank all of you once again for taking the time to join our webinar today. We trust this presentation will provide valuable insight into how the TB11 dual-plasmid system can help your AAV project. So if you have further questions about the data, our platform, or how PackGene can support your specific gene therapy program, please do not hesitate to reach out. So you can email us anytime at the info@packgene.com email address. Before we officially close, I would encourage you to check out our website at packgene.com. So we are continuing expanding our offerings to accelerate your research, including the newly launched, analytical service that we have, like mass photometry for the precise, for an empty capsid ratio analyze, and also most recently the high throughput AAV capsid screening kit, we just launched to help you quickly identify the optimal serotype for your specific tissue target. So please go to our website and check these things out. All right, so we are now closing our webinar, and thank you so much for joining again, and have a good day. Thank you. Bye-bye.
Thank you, everybody.